Hello and welcome to today's lecture on immunity to parasites. Throughout the following lecture, we will be using figures predominantly from your Janeway textbook, and if you would like to reference them later on after the lecture or during the lecture, you can find the figure numbers at the bottom of the screen. Now, pathogens come in a great variety of sizes and lifestyles, and this means that the immune responses to such pathogens is going to differ between the different types of pathogens. Now here you can see the different kinds of pathogens, everything ranging from viruses to intracellular bacteria, all the way through parasites. Now all of these have evolved mechanisms to invade their host, and they all differ in these ways, which means that the immune response against these pathogens is also going to differ. And as you know, immune responses come in a variety of different flavors, both very broad immune responses that target pathogens of a variety of different specificities, and the adaptive immune system, which is highly tailored to the specific pathogen that is invading the host. Now, both of these act together, the innate and adaptive immune systems act together in order to eliminate the pathogen and ensure the health and well-being of the host. And it also depends on the level of the microorganism that is there. And all of these work together, and it's really critical for the well-being of the host. Now, today we're going to be talking about parasites. And parasites are some of the largest pathogens that are known to infect multicellular organisms. And as you'll see, they also come in a very large variety of different flavors. Now, when we talk about immunity to parasites, this lecture will really build on your knowledge of immune responses to show how the different responses work in consortium to eliminate pathogens based on their PAMPs that they might display, or their infection patterns, or their immunopathology, and things like that. So hopefully this lecture will show you that all of the different building blocks that you've learned about immunity are really critical in studying the full host response to a particular pathogen. Now today we're going to go very briefly through some parasitology so that we can get a basal understanding of what the different types of parasites look like, how they act differently, potential PAMPs they could express. Then we'll go through some innate immune responses against parasites, and as you'll see, innate immune responses, although they are critical, the major place where where parasites are really eliminated is during the adaptive immune responses, and we'll go through those. And finally, we'll conclude with some evasion and subversion strategies that parasites have evolved so that they are not eliminated by the host. And this is a really interesting way of looking at immunology because, as you may know, hosts and pathogens really work kind of in a tug and pull manner as the host tries to eliminate the pathogen and the pathogen tries to stay in the host. So let's get started with a brief introduction to parasitology, where we'll go through some very basic general aspects of parasites and then go through a couple different types of parasites. So what are parasites in general? So it kind of depends, but in general, parasites can be termed those organisms that obtain sustenance from a live host, which means that it can be any sort of pathogen that needs a live host to survive. This is a very general idea, and you can probably imagine that any sort of virus or bacteria or pathogen can really be considered a parasite. However, in immunology, there's a slightly more specific term, and it refers to protozoa, worms, and ectoparasites specifically. And so we'll go through what these three classes of parasites are in a few slides. But just keep in mind that the word parasite may have different meanings depending on the context that you're looking at it in. And today we're going to be focusing on the immunological definition. Now, parasitic infections have a really tremendous burden of disease, predominantly through the tropics and subtropics, but also in more temperate climates. Parasites, as they are larger organisms and also can be multicellular organisms, they actually live better and tend to have larger populations in those tropical and subtropical areas, so these regions are more burdened by these types of pathogens compared to other locations in the world. For example, malaria, which causes the most deaths by a parasite globally, had approximately 660,000 deaths per year, mostly among small children in sub-Saharan Africa. So parasitic infections are a really big burden on the world health. And so it's really critical to understand how pathogens infect their hosts, how our bodies respond to parasites, such that we may gain insights as future scientists and clinicians into how to battle parasitic infections. Now let's dive into the 3 types of parasites that are very generally defined. Again, there's a lot of different types of ways to classify different types of pathogens, including parasites, and it again depends on the type of field you're in. So for example, microbiologists may define different pathogens differently based on what they're looking at and comparisons they're making. But for us, I'm grouping types of parasites based on very general, similar immune responses that they can have against them. So the 3 types of parasites are Protozoa, helminths, and Ectoparasites. Now let's dive into some details about these 3 classes. So Protozoa are microscopic single-celled organisms that can be free-living or parasitic. Now keep in mind that you might know of other single-celled organisms such as bacteria. Protozoa are actually eukaryotic, so they have membrane-bound organelles as well as a membrane-bound nucleus. And these are very interesting, and they cause a variety of different diseases. So for example, there are intestinal protozoa, which are transmitted through a fecal-oral route, and you can get an intestinal protozoa infection from contaminated food or water, or from person-to-person contact. Now, intestinal protozoa tend to cause diseases of the bowels that tend to lead to diarrhea. There are also protozoa that infect blood or tissue, and these are transmitted typically through an arthropod vector, such as a mosquito or a tick, as these vectors will directly inject, unknowingly, protozoa into the host's blood or tissue. And these protozoa can cause a variety of different disorders. Now let's look at some more specific examples. So some intestinal protozoa examples include the disease amoebiasis caused by this Amoeba histolytica, which you can see right here. And this is a disease that's caused by this parasitic amoeba. And then when it's symptomatic, it can cause dysentery and in really intense extra-intestinal gastritis and other issues. You can also get giardiasis from intestinal protozoa and cyclosporiasis. And again, most of these disorders are characterized by intense intestinal problems. You can also have blood or tissue protozoa. So here I'm showing malaria, the disease malaria, which is called by Plasmodium, which you can see here. It's this little guy right here. And you can also have leishmania, which is caused by the protozoa leishmania. So these are a variety of different diseases that are caused by protozoa. So then we move on to helminths, which are kind of one of the more frightening, I think, looking examples of parasites because they're very large and they're multicellular organisms that are generally visible to the naked eye. So these helminths, they can either be free-living or parasitic, which means that helminths can live outside of a host, or they can live inside of a host and cause damage within the body of the host. But it depends on the helminth and their specific life cycle. In addition, helminths, because of how they live, they don't necessarily multiply in humans in their adult form. Typically what happens is in the adults, larvae or the eggs of the helminths are planted. These animals will then grow into their adult stages, which you'll see some photos of in the next slide within the human, and then they'll leave the host and replicate outside of it. There are 3 major groups of helminths, including flatworms, horny-headed worms, and roundworms. So if we look at some examples of helminths, we have our flatworms, and these can be generally characterized into 2 major groups. So the flukes, which you can see here. Here I'm showing a blood fluke, which is called a schistosome, which causes schistosomiasis, which again causes a lot of blood problems and some anemia. And you can also have tapeworms, which are much larger. And you have roundworms. Here I'm showing this roundworm here, which is called ascariasis. And these are physical worms that you can see coming out of humans. So you can see them by the naked eye, you can hold these worms, and they live inside their hosts. So the horny-headed worms that I discussed in the previous slide, although they are characterized as helminths, they typically don't infect humans and are predominantly infect animals, so we won't be going through them today. But you can see this very wide variety in types of helminths. So even though helminths are all grouped together as one large type of parasite, you can see there's very different characteristics, and they cause different types of diseases. Now, ectoparasites can be broadly include blood-sucking arthropods such as mosquitoes and ticks and other large animals. And although they are considered parasites and there are specific immune responses against ectoparasites to prevent them from biting larger multicellular organisms, we won't be going through the immune responses to them because in terms of immunity, they tend to be really critical as vectors or transmitters of pathogens. So for example, as I talked about before, there's a variety of different diseases that can be spread through arthropods into multicellular hosts. So this is why we won't be covering the ectoparasites directly, as they tend to spread smaller pathogens, such as smaller helminths or protozoa or bacteria to their hosts. So now that we've gone through a very, very brief overview of the different types of parasites and what they look like, let's go through some innate immune responses to these parasites. Specifically, I want to cover different PAMPs that can be recognized by the innate immune system that parasites carry, and then go through some ILCs or innate lymphoid cell responses against parasites. So parasite PAMPs are very unique, and they actually aren't studied as well as the PAMPs of bacteria and viruses. And you'll see this throughout the lecture. Responses to parasites are really not as well characterized as immune responses to bacteria and viruses, meaning that the parasitic immunology field is really wide open for new discoveries, and this is a really critical burden currently in academia. But you can see here that the different types of parasitic PAMP recognitions is also very limited in terms of our knowledge. So we know that there are some macromolecules that can be recognized by TLR2 on the surface of our cells, as well as the surface of parasitic cells or organisms. But also parasites can be recognized by their very unique types of DNA. So, for example, some parasites have unmethylated CpG within their DNA, which us as hosts do not have. And so if a pathogen were to be phagocytosed, the TLR9 within the endosome that phagocytosed it could recognize that. In addition, parasitic DNA has certain features that allow for other cytoplasmic PRRs, such as cGAS, to recognize it. But one of the major ways that parasites are recognized and immune responses are initiated against parasites is DAMPs. So DAMPs, if you can remember, are damage-associated molecular patterns. So unlike PAMPs, which are pathogen-associated molecular patterns, DAMPs are molecules or molecular patterns that are derived from the host. When a parasite or other wound-causing agent comes through a multicellular organism, there are wounds and damage to tissues. When this happens, certain molecules are released from those tissues that are injured that then lead to the activation of innate and adaptive immunity. For example, a nuclear protein called HMGB1 is very well expressed in the nucleus. However, it doesn't really leave the nucleus in a normal setting. However, when a cell or a tissue or skin is damaged, HMGB1 is released from the cell into the interstitial space, and PAMPs or different PRRs can recognize this DAMP in order to stimulate the innate immune response. And this is why DAMPs are really critical when fighting parasites, as these parasites tend to be larger than the host cells themselves and tend to cause a lot of damage to the tissue that they're in. In addition to PAMP recognition being really critical on the innate side of things, there's also a subset of cells that's really critical in the response against parasites. Now, when we think about the response against parasites, it's very unique from the response against viruses and against bacteria. This is because parasites tend to be very large and tend to be multicellular, or if they're unicellular, the cells tend to be very large and also tend to be larger than the cells of their host. Now, this means that phagocytosis and other means of immunity tend to not function very well. And so the major way of immunity against parasites, the major way that it works, is actually the expulsion of the parasite. So trying to get the parasite to get really, really uncomfortable in its environment so that it either dies directly within the host or it's expunged and removed from the host. So a majority of the immune responses against parasites function to achieve this goal of expulsion of the parasite. So for example, the major category of innate lymphoid cells that acts against parasites are called type 2 innate immune cells, or ILC2s. So this innate lymphoid subgroup of ILC2s is really critical in producing IL-5 and IL-13, which are important in the expulsion of extracellular parasites. And we'll get into why these effector molecules are so important. Now, these cells are induced by IL-25, IL-33, and TSLP, which are produced upon the recognition of the pathogen-associated molecular patterns or damage-associated molecular patterns that we just talked about. Now, these ILC2s are really important because the IL-13 and the IL-5 that they make will stimulate effector responses, which kind of bridge innate and adaptive immunity. So interleukin-5, or IL-5, is really important in stimulating B cell growth. So these are going to be cells that generate antibodies that are specific for the invading parasite. But it also is really important in mediating eosinophil activation. So eosinophils seen here, the effector functions of that we'll get into in a little bit, are really important in the elimination of parasites. In addition, IL-13 is really important in also activating B cells, but it also activates a variety of other different responses and stimulates the production of mucus. And you'll see why that is critical. Now, mast cells, eosinophils, and other cells in these class are really important because they contain granules that contain histamines and other active agents that are really important in the elimination of large pathogens such as parasites. So as you may remember, mast cells and eosinophils and other cells begin their development in the bone marrow, but then they migrate as immature precursors into peripheral tissues. These granules that they have contain, as I said, all these active agents that can be used to expel different types of parasites. And mast cells are one of the most critical of these cell subsets. In addition, we have eosinophils, which are really important in killing antibody-coated parasites. The actions of mast cells and eosinophils we'll get into in a couple slides when we get to humoral immunity, as mast cells and eosinophils act together with humoral immunity to expel the parasites for which they're specific. In addition, we also have basophils, which are really important in augmenting anti-parasitic immunity. However, the actions of basophils aren't as well characterized, and we won't be getting into the specifics, but in general, they also have these granules that are very dense in antimicrobials and molecules that would make parasites uncomfortable in their tissues, and so they're also critical in the innate immune response against parasites. Now, ILC2s, as I mentioned, this is a very general schematic of their actions, but in general, what happens is that upon detection of helminth infection, either by epithelial cells due to damage or recognition of PAMPs on the helminths, the sensing cells will release specific molecules in response to the recognition of the parasite, including TSLP, IL-33, and IL-25, which will activate ILC2s, whose major function is to secrete IL-13 and IL-5, which will not only stimulate the critical innate immune cells, the ones I just discussed, such as eosinophils and mast cells, against this parasite, but IL-13 and IL-5 are also very critical in activating adaptive immunity against parasites as they stimulate B cells to start secreting antibodies of a specific class that allows for the significant elimination of parasites. So now let's dive into some adaptive immune responses against parasites, as I know that I've been hinting to them for a while now. And so today we'll be going through Th2 effector cells, and we'll also go through humoral immunity and B cells. So type 2 immune responses also have an adaptive side to it. So typically when we talk about type 1, type 2, type 3 immune responses, we have our innate side and our adaptive. And since we've gone through the innate side of it already, let's jump to the adaptive side, where the type 2 adaptive immune responses on the T cell side are mostly carried out by CD4 positive cells. So upon activation of the adaptive immune system, naive CD4 cells, when stimulated by IL-4, will become, and when they recognize MHC class 2 peptide clusters, they will differentiate into Th2 cells. So Th2 cells are those cells that are part of the type 2 adaptive immune response. So Th2 cells are also very critical, as they, in addition to helping activate the humoral immune response, they also secrete large amounts of IL-13 and IL-5. Again, this is really critical in launching both increasing the amount of the innate immune response and activating the adaptive immune response against pathogens. But they also secrete IL-4, which also aids in the elimination of these pathogens. Now, in addition to secreting out these cytokines, type 2 adaptive immune cells have a really critical role in antiparasitic activity. So now let's go through some more details of how Th2 cells function in response to a parasitic infection. Now, keep in mind that the major goals in terms of an adaptive immune response against a parasite is to flush it out of the body and to maintain the health and security of the host. So, as I mentioned, Th2 cells produce IL-13, and in addition to them being critical in activating B cells and eosinophils and mast cells, IL-13 is really important in inducing epithelial cell repair as well as mucus production. Now, cell turnover, which in reality is cell repair. This is really critical in terms of a parasitic infection because, as I mentioned, parasites can be very large, so they can cause a lot of tissue damage. So, for example, if you had a parasite that is in your intestine or in your liver, it can cause a lot of cell death and a lot of cell damage, or causing internal wounds, essentially. And the presence of IL-13 allows for cells in that injured area to replicate more readily and to turn over much more quickly. And this is important in two major ways. First, any internal wounds that are caused by a parasite are more readily fixed, and so there's not going to be as much damage, hopefully, within the host. In addition, the cellular turnover will cause milieu to be secreted into that interstitial space, which will want to be cleared by the body naturally, and so there's going to be more things to clear within those areas. So, for example, if we look at our intestinal parasite again, we have our cellular turnover that's allowing for that the potential injuries to be fixed quicker, but it's also secreting more dead cells into the intestinal space, which means our body's going to have a natural response to want to push those out. And as we're pushing out the dead cells that are turning over, we're also going to be hopefully pushing that parasite further and further out of our bodies. In addition, IL-13 that's secreted by Th2 cells increases the production of mucus. And we'll see in the next slide a little bit more about this, but mucus is really important in preventing pathogens from adhering to cell surfaces. So again, our intestines are a very popular example of this, but we also have mucus in our lungs.